Hello, everybody. So today we're going to talk about geneXplain platform, and basically this is it's given for you for free, so you can immediately open it and work and do it a lot of bioinformatics and systems biology tasks to analyze your data. It's a very powerful platform. And I invited today Neetu Tandon. She is our experienced power user. So she's a biologist, and when she needs to analyze data, she immediately goes to to geneXplain platform and use it. So I want her to explain to you the very start of this platform, how to use it, and what are the benefits. At the end, I will also add something from my side. So, Neetu, is your turn.
Thank you, Alexander. I'll just share my screen quickly. Yeah, so geneXplain platform, as most of you must be aware and know that it is an online toolbox designed, developed by the geneXplain team to perform a broad range of bioinformatics and systems biology application. And as Alexander mentioned, the access to geneXplain platform is absolutely free. Anybody can register. So let's quickly see how we can get a free account on geneXplain platform. For that, you have to first go to our website, genexplain.com, and press the login button. You get a login pop-up menu here, page. As soon as you open here, there is an option called New Users, Register Here. So you have to click here, and with this, you can see that there is a radio button activated, registration for a free platform account. So you have to fill the form with all your credentials, generate a username and password for yourself, and just click register. Once your username password is generated, you go back to the login form and click login. I have generated it for myself, so I now just sign in using my credentials. As soon as you sign in, you will see that geneXplain platform product is now assigned to your account. You just have to click on this and the platform opens for you. It looks something like this. So before I demonstrate the functionality of platform, a quick overview of the platform. Basically, the start page. This is also our workspace. So anything which you open within the geneXplain platform, any method or workflow, it gets opened into this space. As you can see here, the start page is divided into a lot of tabs with respect to different types of data. So we have combined different types of methods and workflows for a specific data type. For example, for RNA-seq data. All the methods and workflows present in geneXplain platform has been together put into this tab for ease for the user. So just you have to click on this tab, and all the possible methods and workflows which you can use for RNA-seq analysis can be found in this tab. You can just click on this workflow and the workflow opens in the workspace, but I will demonstrate how and which workflow during the course of this presentation. Just a quick note here is that some workflows are designed exclusively for our proprietary databases, TRANSFAC, TransPath, and HumanPSC, and those workflows will be grayed out for a free user. But nevertheless, like I showed, and there are a couple of other category workflows, and there are a lot of them which are accessible for a free user. The left side, as you can see, this we call it as a tree area. You can see that there are 3 tabs: databases, data, and analysis tab here. The databases tab basically consists of all the databases which are accessible for a free user. And the data tab again contains 3 folders. There is an examples folder, projects folder, and a public folder. Examples folder consists of a lot of examples. So there are some 25 examples which are curated by us. This helps all the users to understand the type of data analysis which can be performed in geneXplain platform, the type of methods and workflows which can be used. It's free for everybody to use, have a look, play around with it. So all the type of files, so you have details about this project, which is explained in this description, which data set has been used for analysis, how this was analyzed. You can open these files from the workspace to the workspace and view the parameters and conditions which have been used. This helps you, gives you an understanding of the tool and the capabilities. After the examples folder, we have a projects folder. So as soon as you create your account in geneXplain platform, you will get your own project created, like a project has been created with my name, and all the analysis which you perform will be under this project. First, in the beginning of the project creation, the project space allocated for the user will be 15 MB. There are two other projects which also you can see. These are our publications, and these are also kind of a demo project, very similar to examples. This project is about the discovery of CpG biomarkers in colorectal cancer cells. Again, you can just have a look. You can play around with these files, open these files and see what all methods were used, how the results have been displayed, etc. Sorry, this is not a good file maybe to open, but yeah, so this is something which we can check. Public folder basically consists of all the background files which users can use for default, but you cannot save anything in the public folder. So these are the files which are by default selected in some workflows, and you can also select it for your methods, but you cannot save anything in the public folder. The next important tab is the analysis tab. So in the analysis tab, you can see that it is divided again into 4 folders: Galaxy, JavaScript, methods, and workflows. Galaxy, some of you must be aware that Galaxy is an open source platform for analysis of bioinformatics data, and we have integrated a lot of Galaxy methods within geneXplain platform that also helps users to get a diverse array of methods and the ease to work with. JavaScript folder basically is the collection of objects for access to platform analysis methods via JavaScript API. So yeah, so then there is a methods folder. So all in all, we have close to 190 methods. These methods have been again subdivided into various categories. So depending upon the type of data you want to work with or you are working with, there is a list of methods exclusively available. So if you want to just work with a track file or a gene table, there are a lot of methods available. So convert table, annotate table, you can form pie charts, bar charts. There are Venn diagram options for data manipulation. Similarly, if you like to work with pathways, molecular networks, there is a set of methods for molecular networks. There is a section for site analysis methods. There is a section, this is a very big section for site analysis methods. There is again a section for statistical methods. So say guided linear model analysis, clustering, Limma, all these methods can be individually accessed by a free platform user. We also have exclusive methods for microRNA analysis. So what all methods we, depending upon the type of data we would like to work on, we can select the method. I would, of course, demonstrate 1 or 2 methods here now. Yeah, so, and now the last but not the least is the workflows folder. Workflows are nothing but they are a chain of methods which are linked together for the ease of the user so that you can perform one-click run analysis. So there are a lot of methods and workflows. Say, for example, in statistical analysis, I have this method guided linear model analysis. This analysis helps me in identification of differentially expressed genes. But if I want to really segregate the genes in upregulated and downregulated, I may need to use another method, which is filter table method. So for the ease of the users, what we have done is that we have made a workflow. A workflow here, I will just quickly show how it looks like. You can So another important thing is whatever you open in the workspace, the description of the same is given in the info tab. So all the methods and workflows, the parameters are clearly defined in the info tab. So it becomes very easy for users to understand and adapt and work with these tools. So if I go back to the workflows and I just show what exactly the workflow contains. So this workflow, for example, it consists of these methods. This is guided linear model analysis methods. Then there is a filter table method. Then there is a convert table method. And all these parameters have been designed and have been calculated by our experts to perform the best of the analysis. However, in some cases, the users might want to change the parameters and they might want to work with their own parameters. And in such cases, there is also an option to edit the workflow. So when you press edit workflow, what you have to do is, say, for example, in this case, the filtering criteria is defined as log fold change greater than 0.5 and p-value is less than 0.05. And I think if I feel that my data, I want it to be more strict, so I can change this value to 0.03. But in this case, this changed form should be saved in your folder. So you will just go here and you will save a copy and you will save a copy into your folder. So just click here and I can click on data here. Yeah. And I save here. So then this becomes my edited workflow. I can play around with it. It gets open. But somehow today I will not use the edited workflow. I will go back to my non-edited workflow. And we will try to analyze this. We'll try to calculate the differentially expressed genes from this workflow. So yeah. If there are any questions or if I'm going too fast, please feel free to message in the chat and we will explain and address your doubts either towards the end or during the session as and when convenient. Okay, so now let us try to calculate differentially expressed genes. For this, we of course need a data. So I am trying to take differentially expressed genes from data from Gene Expression Omnibus. They are TNF stimulated HUVEC and HMEC cell lines. It's a very simple data for demonstration. So what I have done is I have saved the normalized expression data onto my system. So let's just quickly upload it. To upload your data, what you have to do is you have to go here to the data tab. There is an option of right-clicking and import. Otherwise, there is a shortcut to import here. Both works absolutely the same, absolutely fine. Now my file is saved in on my computer. So I just go and I quickly select my file. It gets uploaded. Now it's a tabular file, so it is automatically selected. The type of table is probes Heatmaps, and I specify the species here. Depending upon your table, you can adjust the header row index and the first data row index. If you feel that the first two rows are irrelevant, you can put the input value here as 3. If you understand that your first data, the data which you actually need to work on starts from row number 3, you can adjust these values and you just press start. So pretty much, yeah, so this is my data file, which is open. Now for this workflow, I also need another file, which is called sample table. So I have already uploaded the sample metadata exactly the same way. It is nothing, but it's the description of the samples and how they are treated with TNF stimulated, which cell line, and just the description of samples. So let's just enter the input, fill the input form with the input terms. So to input the form, there are multiple options and drag and drop works absolutely fine. You can just drag and drop your data. It is not necessary that you have to always open your data in the workspace. You can also drag and drop from the tree area. Like I said, we call this area as tree area. So you can just drag and drop from this area. It works absolutely fine. So input table is normalized data, probes is Heatmaps, type of input table is expression values, the method used was quantile, and sample table is sample metadata. So these columns, like I said, so in this case, you have to define the factor which main factor to define comparison. So you have to go to your table and see, in this case, it is the column name is street cell. So I just put this as street cell. You have to make sure that you're copying it correctly. And like I mentioned before, this input parameters are also defined here in the info tab very clearly. So in case you are confused, in case you want more understanding, you can always refer to the info tab as to the input parameters are. The reference level, basically, reference level wants, in case if you want that any of your values should be subtracted from the main value, that type of that is the reference level. But in my table, I have just a none and TNF. So I would just put it as none here. And I compare it to reference, yes. And this is the results folder. Again, the results path has been automatically decided. You just have to, you can obviously alter the names, whichever name you want to put it into, and you can just press run workflow. So the workflow starts executing. Important part here is that whatever happens in the workflow, the details of that is described here. As the workflow progresses, you can read what packages are getting installed. Also, you can check the progress of the workflow in the overview tab. So here you can see that this method is 20% complete. Sometimes it happens that your input table is not compatible to one particular method. In that case, you also get some error messages which are very clearly defined in this workflow. So all in all, it's like a very click and run way of doing it without any programming, but it takes a couple of minutes to perform. So I will just show you how the output looks like to save some time. So once the workflow completes, this is how the output table looks like. You get Limma results. This has down-regulated genes, up-regulated genes. So I have opened, say, upregulated genes here. You also have the list to your TNF versus none results, like we have described here. All these results which you are getting, you can export the entire folder if you want. You can export the individual file if you want to your system. In whichever format is more convenient for you, TXT format or CSV format, and it automatically gets exported to your system. Yeah. Now this table I can also use as a, this table I can also use as an input to other tables, to other workflows, or I can perform other analysis methods within this, with this table. So a very small, minor, important feature which I find in geneXplain platform as very user-friendly is the right-click. Right-click practically does most of the things. So like in this case, I just want this tab to be open. So I just close the other tabs. So this is something very, very useful for me, I really find. Another important thing is whenever you are working on a lot of files and now you don't know where is this file located in your project. So you just have to click again, right click and show in tree. So it will automatically, open and show a tree. It's, very, very useful when you have large amounts of data saved on your project and you want to access and find that file. So instead of searching, you have just one click button and it just takes you to your project immediately. So now I have a list of genes, and what I want to do with this is I would like to demonstrate some methods which I have shown above with this set of genes. So I can maybe use, say, I want to work with molecular networks, and working with molecular networks, I think the prerequisite is to have a genes with UniProt IDs. So let's just convert this table into a table with UniProt IDs. To convert the table, again, it's a very, very straightforward process. And since we have a very small time, it would not be practical to demonstrate each and every method, but the overall schema of working with methods and workflows remains the same. The input, how to import, how to change the names, where you can find the parameters, everything remains the same for all methods. So like here, I want to just use the method convert table. So I had used my gene and I press. So there are different forms in which you can convert. You can change it to gene symbols. You can change it to UniProt IDs. And I want it to change to UniProt IDs. So I just press run here. And as I mentioned, it is like very, very fast forward and very straightforward method. Even when you run the methods, you can see that, you know, there is a clear description of how many rows are matched, how many rows are not matched, when the output is generated, how much time it has taken. So this I feel I have not seen in any of the other products which I have worked on, but yeah, this is something which I feel is very easy, convenient for all the users. So now I have a table with UniProt IDs. So next, possibly if I want to, you know, filter this table, so I can use another function, filter table. Say, for example, I want the list of genes with log fold change greater than 1. So I just have to, you know, use this, and I just have to write the condition log fold change greater than 1. Rows for which expression is true. Yes, I don't want the fit table, but if you want, you know, there is also an option here with highest value, lowest value of expression, but I just want the table which have this condition as true, and I just press run. So it just filters out and I get 77 genes. We started off with 148. So yeah, this is about filter tables. So similarly with tables, there are a lot of methods which you can perform, but I would also like to demonstrate something on a bed file. So I'll close all of this now. Sorry. Yeah. So another example which I want to take is from the gene set. This is again from Gene Expression Omnibus. This is a data set which they want to map the binding sites for SNAIL1 transcription factor in colorectal cancer cells. So I have taken just one of the samples for demonstration, and this time we will be doing an FTP upload. So I have to just click, right click here and copy the link, and I go back to my page. Sorry, this one. Yeah, and I go back to my data here, and again I import. Here, this time I go and I paste my link, and I press OK. So you get a file here, you get the data. Now it's a bed file. So I enter bed file. Sequence source, I think it was HG19. Yeah, the genome build is HG19. So we enter the genome build as HG19 here. Of course, if it is another build, you can select whichever build your data is from, and you have to press start. So your table is uploaded. Let's see. Yeah, so I have my table. Let's just click on this tracks. And we can see that, you know, these are the peaks. This was the table which I have uploaded. This was genome build 19. You can also play around here. You can check the default peak is like this. If you want to have the detailed version with sequences here, on the bottom here, you can see there is the sites tab which is activated. This sites tab will give you the sequence number from which point to which point the peak is there. One particular peak is there. Sometimes it happens that you want to visualize this table not as a genome browser, but as a table. So here again, this is again a very, very minor but very important feature of geneXplain platform. You just open this as table. You right click it and you can see that the entire peaks file is visualized as a table in a tabular form. So for me, like, visualizing in tabular form gives me the correct data. I can compare, I can work on it. So this is also one of the features. Of course, you can visualize as genome browser if you want. There are both options available. Now, here again, if you want to, filter this track file and you want maybe work with peaks only with length 200 or 250. So again, you have methods to work with track files. You have to just filter track by condition. And Again, the same process like I demonstrated for filter table. You have to input your track, put your condition, and you will get a filtered track file. Now, I want to personally run a workflow with this track file. So I will be using the ChIP-seq workflow, and I want to functionally classify this. So for functional classification, I have this workflow. Let's just again look at this workflow once. What exactly is there in this workflow? In this workflow, you can see that the track file is subjected to the method track to gene set, which is then further annotated. And then this gene set is functionally classified. You have different functional classifications to geo-ontologies, to TRANSFAC, transcription factor classification, to Reactome. So let's just run this workflow and see how the output looks like. Before that, now I don't want to work with a G19. So I think I want to work with the latest build. So we can demonstrate one more method from the Galaxy folder. Here there is a method called liftover. So what you can do is you can convert the genome coordinates of your track file, which was a G19 build, to a G38. So we have a list of combinations and we will select a G19 to a G38. And I just keep the mapped and unmapped coordinates as track and track 1, and I just run it. So again, you can see that what method is added, it just takes fraction of seconds to work. So this will be for sure the unmapped part because these are G19 and we will have G38. So this is the file and I should just close all the tabs to, or maybe this tab. So yeah, one moment. Let me just open the output file now. So we have this file and yeah, this is the table, tabular form for just a better visualization. Yeah, in this case, the max peak name is converted to unsure, but nevertheless, the workflows recognizes this as a ChIP-seq peak. So let's just run this file with our workflow and try to functionally classify this. So I had done the exact similar steps which I've done previously, just drag and drop. Annotation sources, of course, it will be with Ensembl human because it's a human data. And then there is a result folder which needs to be created, which you can specify the path and it is automatically created. Again, I press run workflow, the workflow starts working on it. So let's just look at the workflow. I had already, run it just to save some time. So this is how the workflow after completion looks like. You have mapping to different ontologies. You have mapping to Reactome pathways, to transcription factor classification. So if I open, you can see the ChIP-seq peaks are mapped to the geo ontologies. These links are all clickable. They go to the geo accession, to the geo database. Similarly, we have mapping to Reactome. These are Reactome diagrams. If you click here, somehow when I screen share and I click, the system kind of crashes for me. So you can just try it and it's pretty straightforward here. And this is kind of transcription factor classification. So the peaks, the transcription factor genes are classified based on the TRANSFAC classification. So with this, yeah, I would like Alexander to take it forward from here. I would stop sharing my screen and yeah, he would tell you more about.
Thanks, Neetu. Yeah, by the way, don't go away. So we have one question, actually two questions in one paragraph. Okay. And the first question is, how do I upload bulk RNA-seq FASTQ files for the analysis? Can you maybe highlight this a little bit?
Yeah, so bulk RNA-seq FASTQ files you can upload via GEO, the SRA links, and there are, just a moment, I have to share my screen. Yeah, so you can just, if I go back to the RNA-seq tab, you can just load your FTP link, and there is a way where you can, just take the FTP link from your SRA folder, and you can directly upload the entire thing here. There is also an option to import all. So, when you are uploading bulk files, there are many files, you can directly upload all of them. If it's a zip format, it will be detected as a zip format. And I think I answered this question. And what was the other question here?
All right. So basically, yes, right. So you can upload FASTQ files, and then there are a number of pipelines which need to show that you can process it. There's a second question, sorry. Can we do 16S microbiota data analysis using geneXplain software? Well, yeah, maybe Neetu, you can answer that.
No, I think you were.
Okay, so I can, because you are mainly using it for RNA-seq and
Yeah.
eukaryotic data analysis. No, we don't have these sets of data analysis functions to analyze microbiota or so like based on 16S RNA or other things. Actually, of course, we can do analysis. The geneXplain platform can do analysis for individual genomes, even for bacterial genomes. Any fungi genome can be done. But as for this metagenomic analysis, we don't have these pipelines in the geneXplain platform so far. And we have actually a question in the chat. There was one. Is there a history stored for all the analysis conditions that are used? Yeah, that's a very good question. Right, can you answer that?
Yeah. So I will again just quickly share my screen. So yeah, so whatever workflow you are working on, say for example, we worked on this workflow. And again, in this info box, when you can see what input track was used, what annotation source was used, and where is the result folder. So this was a very simple workflow with just 3 input parameters, but this everywhere it is saved, and as soon as you click, you will get the option here in the info box. It is saved completely. Yeah.
Yes, right. And actually, if you click on the link on this info box, it will open for you this form with all the parameters which were used to analyze. So you can actually rerun it again. And if somebody has run it before, you see it in examples, for instance, you click and you get it for you, ready to run it for yourself. All right, so thanks, Neetu, for this introduction. So you are getting, I guess you all are getting now a little bit of introduction of that, and probably you are fascinated to run and try. I will show you a little bit more of that. Let me share the screen. All right, so I have already opened this platform for you, and but before going to that, let me introduce you a website we just created. It's a geneXplain Academy, where you can choose the training. So today we have this coffee break with TRANSFAC, and we are already scheduled another one for next week. So if you have more questions or you need to understand a bit more, welcome to come back to us to the next coffee break. And actually the main idea here of our academy, and basically of our tools of the platform, is we call it no coding bioinformatics, which means you don't really need to learn how to do complex programming to analyze your data. You probably don't have time to learn it, or if there are bioinformaticians which are doing for you, they might be busy. So here you can really do it by drag and drop, by clicking, and save time for for such routine like writing programs. Somebody will do it for you. Spare time for your real research. So that's our motto. And actually, let me go further with this platform. Please put your more questions here in Q&A. I will try to answer them, and or in chat, it doesn't matter. All right, so one important feature of the platform is actually you can share it with other people. And for instance, you've seen this project which Neetu was showing with all the data she generated and results. And I'm sharing this, she shared it with me, and now I immediately see it and I can visualize everything she did and also actually do the further steps of the analysis. So also I can do analysis steps using my own folder. And important is, of course, in this free account, which we are showing here, so you can do everything, although you're a bit limited with the space. So you have to be careful not to get more space, which is offered for you for free. Well, if you get more results, you can, of course, upload them to your computer and then delete the file. Then you can free the space again and run your stuff again. But of course, if you want to do such thing like bulk RNA-seq analysis or and you want to store the results and store the data, then you need more space, and it's easy to get more space here. Just to get contact with us. All right. So and here, now let me just show you some few analysis steps which might be of interest for you. First of all, of course, you might be interested in site analysis. So I guess many of you came here because you know geneXplain as a provider of TRANSFAC database, and maybe many of you just got it there because you want to analyze your sequences, your promoters, to find transcription factor binding sites for various transcription factors. And of course, the best way to do it using TRANSFAC. But if you don't have TRANSFAC, you still can do it here to a certain extent. Of course, also to see what can be found in the first, sketch before you would be perhaps interested in particular transcription factors, and we'll dive more in depth. And for that, you may need bigger libraries. So we have a small library here available for everyone, which is located here in this GTRD folder. It's a set of matrices which are actually built from ChIP-seq experiments and also grabbed from UniProt database. Here we have almost 300 matrices. And plus in ChIP-seq, there is another 128. So we have here a collection of positional weight matrices, which can be used to analyze your data. And let me just show you how to do this. So I uploaded here a file, which is from the same experiment, but where actually I did this selection of upregulated genes treated by TNF-alpha. And here we have these 58 upregulated genes. And now I want to analyze promoters of these genes to detect actually enriched transcription factor binding sites. So for that, you go to the workflow. So let me go here. So you would analyze your genes or protein lists. You just need we started from a list of 58 genes. And then here we go to the analysis of gene regulatory regions and identify enriched TFBS using using GTRD matrices. And it opens for you this workflow. And then you have to basically drag and drop here your set of genes. And by default, it uses housekeeping genes as a control set. Of course, if you have a control set, for instance, you have sets of genes which didn't change expression in your experiment, then you can put here this set as your background. If you then have such a list, you have just your list of beloved genes, you can put here, and it uses by default the housekeeping genes as a default. And here actually, to analyze this, to use your matrices, which are provided here for the free platform. For instance, you can take these profiles, which are called strong thresholds, which means it will search for transcription factor binding sites with relatively high cutoffs. So you find not too many not too many sites. And then you simply run this workflow. And let me see if no, that's one is not available for this type of input. Let me see what I did here. Or no, that's let me. All right, so yeah, the pipeline is actually not working here, but what you can do here is just to work with directly with the analysis function. So here, the pipeline, it would convert everything into easy way. So it would convert your set of accession numbers to proper gene names and so forth. But if you, like in this case, you get an error message because input is not prepared for this pipeline, so you can do it step by step. So you still, it's a little bit of programming in a sense by clicking and running. It's not just one click, but you have to click around. So here we have this method. It's called search on gene set. And here you can do this actually the same. You put, this gene list, and now you have to put your background by yourself. It's not doing it automatically, but it's okay. We can do it, right? So basically, we take this list of housekeeping genes. We put them here. Here you define your promoter lengths from to where. And here in the profile, you have to provide this profile from this collection of matrices which we were just using, and that's it. And then you can run this method, and okay, so it will take, well, it's doing quite quickly. So basically, the output is such a table. We'll not going too much in details. I want to show many things in the platform. Just it actually demonstrates to you which transcription factors binding sites are enriched in your sets of genes, and what is the p-value of that enrichment. For instance, the transcription factors for NF-kappa B, transcription factor binding sites for NF-kappa B factor, they 7 times more frequent in the promoters of these genes, and this difference is quite significant compared to the background, right? So that's how you can use this search using available matrices in our platform. If for some reason you have your own matrix collection and you want to use this platform to run this matrix collection, no problem, you can do it as well. So basically, what I have uploaded here, so basically I had a collection of matrices collected from HOCOMOCO database, and you can simply upload this collection of matrices here, and then you have more matrices, like 400 matrices in your hands to analyze. Of course, TRANSFAC, the latest release has more than 10,000 matrices, but here it's still quite a lot, 400. And the newest HOCOMOCO, I think, has even more. Basically, it would be enough to do your first scan. And when I run this HOCOMOCO analysis, basically now I have much more. No, that's not this MEALR search. That's another program. So the HOCOMOCO results are here. So as you see, I got like 141 positional weight matrices from this collection, which are enriched in my promoter. And here, NF-kappa B transcription factor is found again as enriched and the number 1. But here we're already starting getting some more transcription factors also enriched, which give you maybe a new hypothesis which you cannot get with a smaller collection of matrices. And by the way, if you run the full TRANSFAC, then you get much more matrices to analyze, and much more transcription factors, which can give you an idea what other transcription factors can be involved. Anyway, so basically there are other things, other tools to analyze the promoters in terms of enrichment, and you might be interested in such tools like HOMER and which is quite famous in that domain. And we integrated HOMR in the platform and through the Galaxy mechanism. Actually, as you can see, there are a number of Galaxy tools which are integrated here. And you can run HOMR if you prefer, and it has its own collection of matrices. And so here, for instance, we have results of a HOMR analysis of that, the same set of genes. And the HOMR results are interesting because they are not only using, HOMR is not only using known motifs, like in this case, it found this set of motifs enriched. And again, we see, I guess NF-kappa B was somewhere also enriched, right? These motifs found to be enriched. But the HOMR is also using the algorithm to find new motifs de novo, and that's also quite useful sometimes. All right, so that's all about the motif search. And there are other number of other methods which can be used to analyze motifs. One, there are two important points I want to mention, and that's something very unique for the platform. Here we have a set of so-called tools for combinatorial analysis. So combinatorial analysis means that the transcription factors regulate my genes not just individually, but in combinations. And to find these combinations, there are many ways. And here we provide two algorithms. One is called MEALR. And this algorithm is actually take your set of promoters and takes all the motifs from the library which you are now using, for instance, the small library or a bigger library, and it tries to construct a linear model. It's actually a sparse linear model to be able to discriminate promoters of your interest from all other promoters. So, for instance, I can show you results of such analysis. Let me open these MEALR results on, for instance, on HOCOMOCO matrices, and it not only finds each individual matrices overrepresented, but actually it finds the collection of matrices, namely 78 together. They actually discriminate my set of promoters. For instance, here, right? So it discriminates my set of promoters, and it classify which promoter is belong to my, to upregulated genes, and which promoter is belong to the background. And by doing this classification, it finds actually which transcription factors can work together. To specify it even more, we have this another method which is called CMA, Composite Module Analyst, which is also available for you. And this this tool, did I write it here? I think I run it here, right? For HOCOMOCO. Again, it actually finds even a smaller subset of matrices like this one. It includes just 6 matrices, which together, the sites for these matrices, they are formed clusters. And by forming clusters, they actually really discriminate my sets of genes from the background promoters. And you see here clearly on this histogram, this red bar represent my set of promoters, where these clusters of these sites, and you can see again one of the main sites is for NF-kappa B, but not only, it's accompanied by other sites for other factors, and they together discriminate these promoters from other promoters. And this you can do with the sets of sites which are available in your hands. Of course, if you have a bigger collection of matrices like the whole TRANSFAC, you can discriminate those even better, and you get much better AUC, and your cluster can be much richer, not only including REL and NF-kappa B sites, but other sites. All right. Okay, that's all about the sites. And another very important domain where our tools are working are the domain of pathways. basically, that's one of the unique feature of platform, that we're not only focusing on sequence analysis, on site analysis. So basically, we are on genome level, but we also work with pathways. And this tool combines these two. There are not very many tools available when they combine two worlds. There are tools available analyzing genomes like Galaxy and any other tools, or tools analyzing just pathways. But the tool which combines these two and actually finds the way how to these two types of data and types of analysis can interact with each other. There are not so many of such platforms, and geneXplain platform provides you such possibility. And among these pathway analysis, there are, of course, a very simple pathway analysis, which you just take your set of genes and you map them to existing pathways. And that's what Neetu has shown by mapping your sets of genes on basically, where was it? You didn't, it was somewhere in your results you were mapping, but we can do it also quickly here. So if you want to simply find the pathway enrichment, the canonical pathway, you go to functional classification, and then you choose your geo category, signaling pathways. So let me open which are available, and metabolic pathways, and you drag and drop your sets of genes again here. So where's my set of genes? I specify the species and then run, and it will actually just take your set of genes and map them on different types of ontologies, including pathways. And pathways it takes from Reactome as a free database. So we have here several databases available for you to map, and these are Reactome databases and also HumanCyc. And then in the results of Reactome pathway analysis, you see, yes, right. So Neetu was showing it as well. So that's the Reactome pathways actually shown to be enriched. But another very important type of analysis is so-called network analysis. So what you do is actually you are not dealing with canonical pathways, but you want to analyze your genes, how they connected to each other in the network. And for that, we have this number of tools here for the molecular network analysis. And there are a number of algorithms to find clusters in the network, for instance, to find the shortest, the longest connected chains. Or one of my favorite algorithms is to find regulators. And that's and for the regulator search, what is a regulator? So you have your sets of genes, and then you want to find which of them, maybe one of few regulator molecules in the network, regulator proteins, which can regulate my sets of genes. And through this network, it should go through, like searching for the shortest path. And that's exactly what it will do. So for that, you need your gene list, and you first have to convert it to Entrance ID, because this database we are going to use is using Entrance ID, and the name of this database is called GeneWays. It's a database which is built by automatic text mining, and it includes a lot of hundreds of thousands of reactions. They of course based on text mining, and it's nothing which is manually curated. So for those who wants to use the manual curated network, they can use TransFAC and they get license for that. But if you don't have it, you can use GeneWays as a first approximation, and then you drag and drop it here, your genes. You specify how far you want to go, upstream or downstream, and you run it. So basically, I will. It takes a bit of time. I will just show you results of that run. Let me see. I was here. So master regulators using GeneWays. So it will give you such a table where it finds, well, it finds quite a number of potential master regulators, but it ranks them according to many criteria. So you can really, trust that the top ones, maybe top 10, those ones which are indeed involved in regulating your genes. So you remember we started from like 60 genes, and we can find those top regulators which can regulate my gene. And of course, you can visualize this in such a network. Yeah, so I can show you this regulatory network. Well, it's now showing from right to left. So you can also basically lay out it, for instance, for the vertical layout. Did I click? Yes. And or do some other types of layouts like here. This is a hierarchical type of layouts. So at the top, you see these master regulators, which are pink. And they, through the cascade of reactions, they are actually able to regulate the genes from your set, which are the blue guys. All right, so this is a network analysis, and you see there are a number of tools which can do that. I didn't mention yet the tools for standard statistical analysis. That's, of course, something you would do first, perhaps, to analyze your data, to see how it looks like. And we have a number of standard tools like what you've seen already, this Limma analysis or guided Limma, which Neetu has applied to the data. You can do much simpler things like principal component analysis. So for that, again, the same thing, you start the tool, you need your sets of data. So in this case, you remember we had this normalized gene expression data. So, and you simply drag and drop it here, and you then specify what are the columns you experiment, what are the columns you control, and then actually I have to specify perhaps some more parameters. Let me just run. Sometimes, all right, so sometimes I have to be more careful. I don't have access, right, I guess, to the Neetu's account to store the results in your folder, but I run it in my folder. Just let me show you here. Somewhere there was these results. I think I need it in Limma. Sample, yeah, PCI, right here. So I run it, and you see this PCA plot, standard plot, when you get these dots for the experiment, and another color of dots for the control, and then you can clearly see whether they discriminate or not. So it's a simple example, but if you have a much more data, these dots can be really crowded. So there are more other tools to do similar things. For instance, this Heatmap is a very nice tool. You really can show the standard heat map, the same set of data. You also see here t-SNE or other tools. All right, so I have a question in my chat. Actually, two questions. First question, is there history stored? That's we already were talking about. History stored for all the analysis conditions that are used, so that Neetu has answered already. Yes, we have. So you have it in an info box. And the second question, do you have any tools for combining technical biological replicas in the single data set to work with? All right, so that's a good question. Well, actually, there are not so, we are not very special in that. So there are standard approaches to do that. And the major one, which Neetu shows, is actually guided Limma, which we have here. And here you see this, this is a main factor, which just shows you which is what was test, what was a experiment, was what control. But there are also other, other columns which can be used here, which can be actually used as additional factors. Like you can apply this, surrogate variable analysis, or you can apply more robust way of analyzing your data, and in that way you actually can combine, as was in the question, whether you can combine different types of data and can control this experiment. So, for instance, you did your experiment in 2 different batches, right? So you may expect a batch effect. Then here in this guided linear model, you can specify which of the data sets coming from one batch and which are coming from another batch. And then this tool will automatically try to diminish this batch effect. And actually, SVA generally, even if you don't know the batch effect parameters, so for some reason, you don't know how to control it, you can apply this SVA, and it will try to find any kind of a batch effect and control it. Of course, you have to be careful, because all these batch effect controlling mechanisms, they can kill the signal. So if you really want to put everything in your analysis and control all parameters, then maybe you'll find no differentially expressed gene, because too much noise in your data, and it was all diminished by the batch effect control. All right, so that's how you can do this a bit more sophisticated statistical analysis. And as again, coming back to the overall schema, the platform is for you to analyze many different types of omics data. I didn't show you some features. For instance, you can even analyze metabolomics data. Just little since I did this, if I can find it quickly. All right, I think this is a type of visualization. No, that's not the one I wanted to show. Where is it? So you can actually do the metabolomics. I think this was one which I wanted to show you. All right. So if you do the metabolomics types of analysis, so basically you have in your hand metabolites, and then you know expression of these metabolites in your experiment and in your control, then to build chains between metabolites and to find how metabolites are connected to each other in a human metabolic network, we are using here 2 types of databases, like Recon database. It's reconstruction of the whole metabolism for human, or another database, HMR. So you can build such networks between metabolites and even give colors, expression values of metabolites on your map. And not to miss to mention this, you can actually save any of these data you generated here in the platform. For instance, such nice diagrams, you can save it for in PNG for just for putting in your publication, or you can save it in a special format for Cytoscape. And then if you have a Cytoscape and you know how to use it and in your Cytoscape analysis, you can immediately import it and do further analysis. All right, so with that, I want to finish for my main presentation. The actually session is still open for your questions. For instance, maybe you're interested in a bit more about the programming access to this tool. So although, as I said, we're promoting this no coding bioinformatics, so which means you don't have to be a bioinformatician to fully analyze your data, and you don't need to wait for bioinformaticians until he find the time to analyze your data. You just take your FASTQ file from a machine, and you get until you find your master regulators, you find your drug targets. So all you can do here, and then All right. So, but there is also a big part of this platform, which is quite useful also for those who do programming, because we have API, and so any of those tools and functions can be accessed through the API from inside your program. So for instance, you write your program in R, and then you want to analyze transcription factor binding sites enrichment in your sets of genes. You just run it through the API and to our platform, and you get results inside your tool, and you do whatever you like with it. So that's you can all do. And for this API, we'll have a special new coffee break in near future where our API specialist will tell you more details how to apply and how to use these API functions in your program. All right, so thanks a lot, and see you next time.